We can start the discussion.
So Adam, actually, I mean, you showed some of our preliminary data that the phenotype gets better. The problem is the function doesn't really. And that seems to be strongly dependent on the duration of infection. And that's obviously a big problem for HBV, where we usually look at patients who have been infected for a long time. And I know Robert has the same data that really shows that. And that's also a caveat. Phenotype is not function, even if they look in some ways actually quite good. And certainly a lot of things change, so that is correct. So, and I think we need to define that better for both HCV and HBV, kind of, where do we get better? In the roadblocks that remain are those things that are druggable or we can interfere with or not. And I think that's a little bit my concern that even if we get some improvement on its own, it certainly will not be enough, I think.
Okay, was there a question? So I will agree with you 100%. I know there was more data than I presented for the Hep C.
It was more a comment because. Another comment, I fully agree with you. We just need to do these studies in the humans. I mean, this is.
Yeah, I know I went light on the Hep C data. That I probably should have put more because it's really our human data, but I know a significant amount of it still is coming down the pipeline in terms of publications, everything. So, but I think it's important to really follow this closely. I mean, the antigen burden isn't as high in Hep C. It's a very different disease overall. There's a lot more variability, but. It's getting rid of a chronic infection, and this idea of the duration of infection, it was alluded to earlier in the other session as well for hepatitis B, not knowing when these patients were affected. I think all these are excellent points that really have to be at least considered in the data analysis for some of this.
And obviously the beauty of HBV is natural history indicates that it is possible that we get immune control even in patients that are not treated. So.
Thanks.
Stefan Urban.
I'm afraid it also will not become a question, but it's a remark to Jordan's talk. I like it very much. I just wanted to add two things. First of all, you find integration in vitro within 3 days.
Yeah.
So double-strand linear DNA, whenever virus is there and a susceptible cell is there, will go into the liver and with a very high frequency integrates. So it's not a very low event. The other comment is regarding the possibility of S discriminating from integrate, coming from integrate or from cccDNA. We've investigated Delta patients, and we especially investigated the infectivity of the Delta virus in those patients by using a TCID50 test. I mean, if you measure delta RNA, we would assume that these RNA-containing particles are infectious, but we found profound difference in the infectivity compared to the levels that you measure by PCR. 3 log of non-infectious particles which are present and which correlate with what you showed by the thing. So we have to assume that maybe delta in e-antigen negative patients uses mostly the cells that have integrate and affords to have non-infectious particles in the huge amount produced, telling you that this comes from integrates. Of course, delta is not a good marker to test the integrate because as I said, but in the delta patients, we have very much evidence that this comes from integrate mostly.
So can I ask a question to both of you? So what is known about the requirements for a cell to become integrated? Is it related to the status of the liver cell? And can you give the microphone back to?
Can you give back to?
I can also speak.
No, let's see. Because presumably it has to do also with the status of the chromosome.
It's a very important point.
Yeah.
This double-strand linear DNA comes in by an enveloped virus, so you can completely repress it with an entry inhibitor. Mm-hmm. So you need the receptor in order to do that. And this is an important point, it probably only can preferentially target cells that do not already express antigens, S antigens. Mm-hmm. So there is a selective superinfection exclusions for HBV that comes into cell that's already infected, because if the L protein is present, nucleocapsid cannot travel to the capsid, to the nucleus. This is also something we should, the reservoir of the S antigen integrates could lower the reservoir of replication space in the liver for B, but not for delta.
I was also thinking of the status of the infection and whether it requires activation of a liver cell to become infected. So my question is, if you look at the dynamics over time, is the likelihood of integration much higher during the acute infection? And do you actually build up a huge reservoir that degrades over time? Or is the model that you have continuous replenishment of the cells?
What we see, I mean, we can also only test it in vitro, but what we see is that most of the integration occur with the primary infection.
Right.
If you make a long-term infection, there is not much of an amplification going on. So the double-strand linear DNA comes mostly from the enveloped incoming virus and not from something that's produced over the next 10, 12 weeks.
Stefan, do you have evidence that the status of, let's say, cell proliferation is important or not? I mean, you can integrate a completely quiescent G0, G1 cell, or I know that you can, but it's important that you...
No, it's not. You don't need cell proliferation for that
So
effect.
apparently you can have an integration in a cell that is, let's say, G0, G1 without any difference, or you have an increased integration when you have the cell that is proliferating.
No, you don't need the
you don't need it is something, but you have an increased round.
Yes, of course. That's the big question, whether those cells that are hit then have an advantage to proliferate. I would assume that we don't know that exactly. It depends on where it integrates genomically, whether it makes a benefit to the cell to proliferate. Yeah, but at least it's not counter-selected, obviously.
Okay.
Yeah.
Bruce, and after we go back to the immunology strict proper. Let's have a little bit of virology, hopefully.
Yeah, maybe again more comment than question, but also I think maybe we'll stimulate some questions. First of all, I think it's sometimes missed that the E-positive chimps, did have integration. It's just a lower percentage of their mRNA is coming from integrants, and I think that the notion that E-positives aren't producing S antigen from integrants, I think is probably incorrect. It's just that the proportion is much, much lower. So you can lower them several logs before you hit that integrant floor. And I think the other thing that we sort of saw, very few patients, of course, but very well studied, is that whether you're an E antigen, that you drive down far enough that you can no longer see core and of course say E seroconvert, and their RNA is also undetectable and their DNA's very low or undetectable. E positive or E negative in that state, they could have S antigens anywhere from a couple thousand to 5. And I think that's just a function of how active their integrated RNA is. DNA is, transcriptionally active. So getting to Jordan's point about, why does anybody clear? We've learned so much over the last few years, especially from the work of, Gilead, about the importance of X protein and core, for the activity of cccDNA. I can't find a lot about what transcriptionally controls integrated DNA. And I don't think we're clearing, those patients that clear, I don't think we're clearing their integrants. I don't think we're killing all those cells. I think, the body is, the host has figured out how to transcriptionally control that. And my recommendation to the field is start paying some attention to what's transcriptionally controlling integration. I recognize how hard that is because it's all over the place, integrating at all different sites. But my guess is that's where the clue is.
If I can turn this virologically driven comment into a question, just so I can give an answer or another comment, and after we go back to the immunologist. I think that what you said is very important. I actually noted something during the presentation because everything that integrates can integrate in an open chromatin, closed chromatin. Actually, for integration, we suppose that you need a more open chromatin, but that does not mean that it will stay open. So we need to know in the future where the integrants are and correlate the integration with the epigenetic status of the regions in which the integrant is. We can do that easily by looking at transcriptional activity, by looking at the number of fusion transcripts that come out as compared to the fusion DNA burden. But this is not easy to do because normalizing all this in a study would be a nightmare. But this is the way to go to understand. But clinically speaking, you have the long-term treated patients with NUC, even with lamivudine, and you have some patients that lose S, obviously, but you have patients that go down, and after they just ramp flat at 155 units and never move. If you try to correlate that with the amount of S-containing hepatocytes, you have a rough correlation, but it's not very, very, very strong. And if you try to correlate with the clonal expansion in the Kennedy paper, there is not a clear-cut correlation between the size of the clonal expansion and the number of clonal expansion and S production. Just to reinforce the concept that whether we believe or not that S is the only determinant of exhaustion and recovery, that is for the immunologist to answer, what is really important in playing with biomarker is really trying to understand this kind of thing. That will need a lot of liver samples. That is the bad news for all of us, whether in the academy or in the industry.
Okay, I'll just confuse you by starting with a virological question now.
Oh.
No, just wondered, so the PCR of the RNA transcripts, how widespreadly available will that be? I mean, does that need a specialist lab to do? Or
What
Transcripts.
For the RNA.
The PCR
HBV
RNA.
PCR for the circulating RNA?
Yeah.
So there are a number of homemade methods that you can replicate. There are a number of labs that did a lot, so you can collaborate with. And there is one test that is not yet released, is not yet is just a prototype in Abbott. And there is a second one that is, I would say, is slightly less developed, but ramping fast from Roche.
So you see this as something that will be available in most
I think that
virology
something
labs,
that can be used in collaboration with Abbott, I think, just now. And with Roche, you have to ask them, but will not be a lot of time. I mean, I would say end of the year, probably, unless there are. But there is a prototype in both companies. There is a prototype, and some of us are involved in studies to just do that. Harry has been involved.
Massimo, you mentioned that the problem with correlated antigen in the negative patients is many patients are negative, and I agree. But that's also true with HBV RNA. Sure. So what do you do with that?
The big advantage is that RNA is still there in the NUC-treated patients. So the niche of the NUC-treated patient that is what you have in real life in the clinics. You can tackle with RNA, and you cannot really do almost anything with the correlated antigen. So there is a problem of sensitivity. That is a sensitivity problem mainly with the homemade tests that are at 1,000, 2 logs, 3 logs. The commercial one that declare a limit of detection around 100, or so is 2 logs sharp, and that might recover some positivity. But large cohorts to be done. The published data from Abbott are on 100 untreated patients and 16 treated patients. So you know it's not a big stuff. They have more. They have much more, but it's not published yet.
Sorry, can I just follow up with a quick immunology question for Adam? So given the fact that the existing HPV-specific T cells have this epigenetic imprinting of exhaustion that might not be fully reversible, what do you think are the prospects of instead inducing de novo responses? So bringing down antigen, using a vaccine perhaps to induce new responses which are then protected from going down that same route of exhaustion?
I mean, I certainly think that inducing a de novo as long as it's going to match the virus that's infecting the patient, if you can match a genotype where you can match these epitopes enough to get actual recognition of the infected hepatocytes, That's what it's going to take. I mean, in the mouse data also shows that if you're inducing proliferation in these T cells, that's when they restore function. And there's more than just the study that I showed here. So if you're actually getting induction of some replication of these exhausted T cells, they do recover function, at least transiently, and it depends on maybe how many times you have to boost. There's been other studies trying to redirect functional immune responses for like flu or things. I think has been shown too. Getting a functional T cell response, whether it's de novo HBV specific or otherwise, is going to be challenging. But I think this is our best. It is a good opportunity to try and get control of the virus.
If there is nobody else, fast, I have an immunological question to all the immunologists collectively, because you mentioned in different ways the fact that you have a kind of phenotype that is imposed, an exhaustion phenotype that is imposed, that you can eventually try to counteract with the checkpoint inhibitors, whatever you want to use, or can functionally, even if it's not a checkpoint inhibitor, you can functionally retrieve the functionality of the cells. But you also mentioned that there is a reduction of the pool of the cells, and you have to re-expand. So you have to have a vaccination. Do we have an idea how much important is one mechanism and the other mechanism in the different categories of hepatitis B patients? Let's say early patients, late patients, in terms of need for re-expansion, need for vaccination, need for as compared with just a simple checkpoint inhibitor to be given.
Did you read our HBV paper that we published?
Yeah, sure. I am allowing you to talk about it.
No, it's a huge question. I think the different mechanisms at different stages of diseases. Yeah, the data so far on the magnitude versus functional recovery ex vivo is still all quite weak, to be honest. I mean, there's the suggestion that there's no real difference in the T cell frequency among these different categories of patients, but you're looking at only a few responses in a few patients in any case. And Carlo's presenting some data now looking at different phenotypes correlating with different cytokine productions and maybe trying to target some of these memory cell populations. And I can't remember the exact details, and maybe Mala can help fill these in on how those changed based on the categories of patients in terms of viral control. But
do you think it's important in terms of strategies for cure? Or, because that is the big question. The seminar is about cure.
Yeah.
Immunologists don't take the bait. Okay. So, Eric, it's up to you now.
I'll come back to the testing of correlated antigen and HBV RNA. Very nice talks. We've tested all these parameters in our series of patients treated with nukes and interferon, and in the E positives, it's pretty easy. If you take S or quantitative E, it all nicely correlates with response, and correlated antigen is basically E antigen almost. It's a very high correlation. It's all driven by E antigen. In the E negative population, it's much more complicated, and there these tests might be more interesting to use, I think, both HBV RNA and correlated antigen. But we suffered, and as Jordan already mentioned, with both tests that the sensitivity is not good enough to really make them useful. So we see definitely for HBV RNA 30% being negative already at the start of treatment, and then even with the higher sensitivity test as developed by Floria von Bommel, and I think it's not much different from what Abbott might have, you see very quickly that the vast majority of the patients are HBV RNA negative, and the same holds true for the correlated antigen. So what is the first question is, to what extent is it possible to increase the sensitivity of these tests quickly for E antigen negative population? The second question I have is, how do the drug developers and also the FDA have to handle these tests?
For the FDA, it's very difficult to say at this stage of the knowledge, at least from my perspective. I would say that would be really difficult to give advice or to discuss, because what they usually want is something much more solid of what we have. Let's say in this way.
But the one caveat to that, which I mentioned, would be to go back to some of the old natural history studies and actually prove that these markers are predictive, because I think that's what we don't have. We know that they correlate with very short-term outcomes, but if you could show that they...
Predictive of what?
What's that?
predictive of what?
Of HCC and survival. Really, that's at the end of the day what the FDA wants, and that's why they use S antigen as their marker, because they've shown.
So we need to reveal data out of Taiwan basically to see whether they, because that's your golden standard at the end of the day for all of this. It's death and HCC.
But going back to the other question, in the case of a correlated antigen, I think that the real point there to have a test that can be more useful is to eliminate the E component into it. Is it just a question of how many antibodies you put in the test? They standardize in that way. They are not so much willing to go back in doing all the work to validate without the E epitope antibody. And you can have a test that just measure circulating core. Whatever is the core. And that would be probably a completely different test in terms of meaning. And this is something that an academic lab will never do, as is in the hands of the diagnostics branch that can eventually do Fujirebio or others. I think that in that case you might be quite sensitive, and you can push sensitivity, because the problem with the Fujirebio is that you can go to 2 logs, but it's not quantitative anymore because the test has a lot of detergents and things inside, because they come from the early steps of disruption that they have to do. So I think that in the hands of good diagnostic tools developers can be done something. For the RNA, I really think that the big point is to know what we are dosing and to know how much confounding factors come from something that is not the pgRNA. Because our assumption is that what is circulating is in the particles, because it has been found as pgRNA, and if it is pgRNA, it's probably inside a capsid, and either is a circulating capsid, or is immature capsid circulating, or is a particle with RNA inside. And this is the assumption, but we do not know exactly. And the other point is to know how much of these transcripts are really spliced. I think that if you have a strategy that can differentiate what is there, you can pick up the components that will be more meaningful. Obviously, in the patients that are treated for a long time, probably you will not have the results. With the test, you have, you say 30% that are negative before treatment. I would say that you can go a little bit down, but you will always have in the E negative population patients that will have a very little things going around. If you look at cccDNA, I know everything comes from the integrated, but still the replication comes from the cccDNA, even in the E negative, and the reactivation comes from the cccDNA. The amount of cccDNA that with our not good tests we can measure is 0.1 per cell or 0.01 per cell in some categories of E-negative patients. So it's very little amount. And this means that you will always have categories of patients that will not have the marker. And then you can correlate negativity with outcomes. And this will be anyway important.
But I think I mean, I know you've studied this a lot, but I do think it speaks to the fact of having a better understanding of what happens with ELOS, because if ELOS is all virally driven, if it's just pre-core mutation or BCP mutation and you don't produce it, that shouldn't really affect cccDNA transcription on its own. So really understanding, but it seems to, we clearly see a drop in, you've shown it, lots of people have shown it, that everything goes down with E0.
The relation between true cccDNA transcription measurement and the basal core promoter or the E- mutations, I think we do not know yet.
I agree.
We are actively studying that with Maura Danbury and the mice infected with the different strains, and we are finding something that is a little bit surprising and not really exactly what was easily done in the patients in the old days with the old tests. So I think we have to wait for that to know more.
No, that's true.
But you have also E-positive patients that do not have the pre-core minus. So they do seroconversion without the pre-core minus. Non-D genotype, they don't do that. Right. So non-D genotype are the majority of the infection in the world.
I'm afraid we're not going to solve this one. I think in the interest of time, we have to stop this session. I'd like to thank my co-chair, the speakers, and you for excellent presentations and very nice discussions.